morning. Myself Dr. G.D. Gupta, Director ISF College of Pharmacy. As you know that this institute was established in 1984. We are running all the Pharmacy Council of India and AICTE approved courses in pharmacy. At present we are running Diploma in Pharmacy, B.Pharm, postgraduate course M.Pharm in 6 branches: Pharmaceutics, Pharmacology, Pharmaceutical Chemistry, Pharma Analysis, Quality Assurance and Pharmacognosy. We are also running PharmD program and PharmD PB including the B.Pharm practice. This institute is approved by the All India Council for Technical Education, Pharmacy Council of India, Government of Punjab and affiliated IKGPTU Jalandhar. This institute NAC accredited A grade college as well as recently this institute recognized by the UGC as a autonomous college as well as affiliating university also approved this institute as a autonomous college for 10 years. In this institute 14 states faculty, near about 70 they are working, and we have 10 professor, 21 associate professor as well as 39 assistant professor in different disciplines. Our research team is working very hard work in the various fields including the pharmaceutics, pharmacology, pharmachemistry, pharmacognosy, quality assurance as well as we can say one of the SPAL lab we are running in house, and in this SPAL lab this is the NABL accredited. So we provide the practice as well as the training to our student in house as are near to the industry. In this institute we have the one of the big pool of the research. Our research team in the 5 year published more than 1,000 research paper, and total impact factor of this research paper 912. We have also published 48 books, 27 we have the MOUs as well as we have the 9 transfer technology. In this institute 18 state students are the studying in various courses. Our management provide the excellent infrastructure, facilities and equipment for the research and the academia. At present we have the good specialized lab known as the nanotechnology lab, plant tissue culture lab, animal cell culture lab, as well we have the nanotechnology lab, one of the most premier lab in this institute. This institute is also running one of the SPSS animal house with the breeding and trading facilities. We have the business incubation center approved by the MSME. Overall, we provide the excellent training to our graduate, postgraduate, diploma student, as well as this institute is also approved as a research center by the university. Good morning. Myself Dr. G.D. Gupta, Director ISF College of Pharmacy. As you know that this institute was established in 1984. We are running all the Pharmacy Council of India and AICTE approved courses in pharmacy. At present we are running Diploma in Pharmacy, B.Pharm, postgraduate course M.Pharm in 6 branches: Pharmaceutics, Pharmacology, Pharmaceutical Chemistry, Pharma Analysis, Quality Assurance and Pharmacognosy. We are also running PharmD program and PharmD PB including the B.Pharm practice. This institute is approved by the All India Council for Technical Education, Pharmacy Council of India, Government of Punjab and affiliated IKGPTU Jalandhar. This institute NAC accredited A grade college as well as recently this institute recognized by the UGC as a autonomous college as well as affiliating university also approved this institute as a autonomous.
Hello, welcome to all the participants for this online lecture series by Dr. Amit Sharma, Head Professor, Department of Pharmacy Practice. On behalf of ISF College of Pharmacy, Moga, Punjab, I welcome all the registered candidates, participants. This lecture series is going to be on biostatistics and research methodology, and the timings will be daily from 11 a.m. to 12:30 p.m. It is starting today from 12th of May 2020. The study material will be shared on SlideShare, as well as you can view daily lecture on YouTube live of YouTube channel of ISF College of Pharmacy Moga. For viewing the lectures successfully, you must subscribe to the channel. This is very first kind of initiative taken by ISF College of Pharmacy jointly with Dr. Amit Sharma, who is a well-renowned expert in biostats and research methodology. Dr. Amit Sharma is having a vast experience of more than 1 decade in pharmaceutical education and research. He is a prestigious alumni of Al Amin College of Pharmacy, Bangalore. He did his Masters of Pharmacy Practice from Al Amin College of Pharmacy, Bangalore. Right now, he is working on the prestigious post of Professor and Head Department of Pharmacy Practice, that is PharmD and PharmD Post Baccalaureate. He completed his doctorate in pharmacy practice from Uttarakhand Technical University. He is having vast experience of 13 years and having more than guided 61 students of BPharm, MPharm and PharmD thesis. Dr. Dr. G.D. Gupta, now I would like to call our worthy director of ISF College of Pharmacy, Dr. G.D. Gupta sir, to come over here and throw a specific light on biostats and research methodology and overall curriculum to be covered during this 10 days online lecture series. So over to Dr. G.D. Gupta sir. Thank you.
Good morning, everyone. So, as you know that ISF College of Pharmacy Moga continue participating and try to resolve the problem related to lecture and related to the problem of the syllabus. So, our team, we all have decided to continue some of the lecture series, and one of our faculty member, Professor Amit Sharma, he submit a proposal. As you know that most of the courses, you can say BPharm, PharmD, MPharm, PhD course and all subject of the M pharmacy, they required some of the kinetic and somewhere of the part of the calculation. It was observed in most of the institute, only a few teachers, they are delivering the lecture on biostatistics and research methodology. As you know that number of the institutions, number of the colleges, number of the scientists, they are available on webinar, available on the guest lecture. So all these lectures are only a few. You can say specifically 1 lecture, 2 lecture or 3 lecture. But we have decided to complete the syllabus, and this syllabus related to biostatistics and research methodology. This syllabus prescribed by Pharmacy Council of India, and in throughout India, in all parts, in B pharmacy and also in the M pharmacy, this course is compulsory. Number of participants from 18 countries, they are registered for this course. So I can say that it is not essential that this is the same curriculum in India as well as abroad, but 90% of content applicable in throughout the worldwide in pharmaceutical sciences. I hope during these 12 days you can enjoy, you can contribute in this subject. So the mode of the presentation, this is the live and available also on YouTube channel. So if you want to ask any question related to this subject, related to the topic, you can send your queries through the YouTube, or you can SMS to our already is the given phone number, WhatsApp, so that we can justify your queries. Every day, Professor Amit Sharma, he will take 10 queries in the morning time. Today's lecture, if you want to know anything, so you can send your SMS or WhatsApp or email. What do you want to know? So that if you have the repeated queries, let us consider 50, 100 or 200 of the queries. These queries sum up and find out how we can summarize. So our team will provide the justification and clarify your queries every day in the morning when we start the lecture at 11 o'clock. So we provide the 10 to maximum to the 10 minutes. It can be clarified, and then as per the schedule, the lecture will be continue. So I have already here the mention that 12 days our team and Professor Ramesh Sharma try to complete this whole of the series. So I request to all listen carefully, and all slides shared on the screen, and this content is also available on our YouTube channel. So connect in timely. Every day we will start sharp at 11 o'clock. So after the 11 o'clock, you can connect with me up to the end of this lecture, so that you can find out so many good things. And you can also include, if you have some of the excellent idea and excellent opinion, or you can say some of the good methodology, this is the required to be incorporate, you can also send it, because ultimate goal of this series to provide excellent content and up to the mark content to each and every participant. Without delay, I will just invite today's lecture series speaker, one of the very dynamic leader, technocrat, and he is the expert in biopharmaceutics and research methodology. So now I request Mr. Amit Sharma to continue. This is the first lecture of this series. So from ISF family, I congratulate to you and give the lot of good wishes for the successful of this series, and no doubt it will be the benefited to all of you. So thank you very much. Listen carefully, and if you have any queries, any question, please connect to me every day at 11 o'clock to the till the end of, and most probably we will try to complete at 12:30. So thank you very much. Now I connect to Mr. Professor Ramesh Sharma, please.
Uh, Welcome to all the viewers. Today, I'm going to start a lecture series of biostatistics and research methodology. First of all, I would like to thank the management of ISF College of Pharmacy for providing such a wonderful uh, platform for conducting this lecture series. And I would like to uh, hello, yeah, I would like to Thanks to our worthy director, Shri G.D. Gupta sir, our chairman, Shri Praveen Garg, our technical coordinator, Mr. Saurabh Kose, and Mr. Paneet Kumar for conducting this session. So I'll just share my screen to you. In today's lecture, we are going to see the four different topics. The first one is descriptive and analytical statistics. The second one is introduction of statistical software, that is SPSS. We are also going to discuss different variables which are used in research and how we will do the coding of SPSS software. We will also discuss the different level of measurements used in the research and the mother of all the tests, that is test for the normality distribution. So the today's presentation is going to help the all medical professionals like pharmacists, nurses, clinicians and all. Before I start, I just want to let you know that data which we will generate, for example, if you're collecting a data from the hospital or from our laboratory experiments, that is classified into two different types. The first one is descriptive statistics, descriptive type of data, and second one we used to call as analytical type of data. So descriptive statistics is the main objective here. In this descriptive statistics, what we used to do, we have to summarize all the variable one by one. For example, if you are going to write a thesis, so during the introduction part of your results, what you are going to do, you have to describe the variables one by one. So how you are going to describe the variable, you have to write the descriptive statistics of all the variables. We will discuss the different variables in the further classes, and today a little bit introduction I am going to give you. So once you will complete it with the descriptive statistics, you have to apply the different statistical test to explain your research. For example, I am going to take that A is causing B. This example I am going to take for all 10 days. For example, A is the smoking and B is the cancer. That means smoking is responsible for the cancer. If you want to justify this thing, so A will be taken as an independent variable and B will be taken as a dependent variable. So in descriptive statistics, you have to explain what do you mean by A. And in analytical statistics, you have to correlate that how A is causing B. That means how your cancer will be caused by a smoking. So in descriptive statistics, the main objective is to summarize the variable one by one. You are going to summarize that what do you mean by A, from where A is taken, how many cigarette is smoked by a person in a day. Clear? And in analytical statistics, we will find a correlation of A with B. So both we have to explain one by one. In descriptive statistics, the first thing to do before we analyze the data, describe the variable one by one. For example, I have taken, if you are doing a study on a hypertension in a population, then we should first describe the population. For example, which kind of population is suffering from the hypertension? Either they are male, either they are female, what was their age group, uh, either they are literate, illiterate. So commonly, if I say, it will be called as a demographic profile of the population or demographic profile of the patient. So when we are giving the description of the population, it can be age, gender, race, education, or income level. So all these things will be the demographic profile of the population. And whenever you are going to write the explanation of these things, this will be called as a descriptive statistics. So when we have to explain our descriptive statistics, we have to choose some level of measurements. Clear? So level of measurements which we are using in the research are mean, median, interquartile range, like that. So how to choose that which level of measurement or measurement of central tendency is the best suited for the data or for the population which you have chosen. For example, if you have taken a numerical data, we have an option for the mean, median, and mode. And if you have taken the categorical data, we have an option for the frequency and count. So there is a different rules by which mean you can choose that which level of measurement of central tendency you are going to use with your data. Clear? Most of the researchers, what they used to do, they used to refer a research paper of some other authors, and if some other author have written their data in the form of mean or standard deviation, what the you pupils or other researchers they will do, they will also write their results in the form of mean and standard deviation. So it is not like that. You have to choose the precision of estimates. For example, you have to apply a test to know that which level of measurement is best suited for your research according to your data which you have generated through your research or data which you have taken from the hospital. So if you want to explain the categorical measure, for example, gender, where you have two different categories, the one is your male and second one is your female. So for that, you are going to use the frequency, you can use the count, and you can write the percentage. For example, 60% male and 30%, 40% is the female. So this example I have already given to you, that distribution of gender, which is a nominal measure, we could describe the number of male and female in the form of percentage. Clear? So whenever we are writing a research paper, in the research paper, it is not necessary that we write the percentage of both. For example, you are going to write a percentage of male and you are going to write the percentage of female. If you have explained the one variable in your research paper, it is going to describe other will be complement the value of other variable. So just describe only one variable. Just for example, you are writing a percentage of male, so no need to write the percentage of female. It will complement the other value. Now, whatever I told you that was about the categorical variable. What will happen if we have a numerical type of data which you have taken in the form of numbers? It will totally depends upon the normality distribution of the data. If we find that our data is normally distributed, we will use mean and its dispersion value standard deviation. Clear? When the data is not normally distributed, we should not use the mean, and we are not even eligible the mean on our data. In that case, we have to use the median and the interquartile range. Interquartile range means data is divided into 4 equal parts by 3 different intervals. For example, if I take the example of 100, so 100 is divided into 4 parts by 3 different interval, 25 each. Clear? So if data is found normal by applying the test called test for normality distribution, we will use mean and standard deviation. If data is not normal, we will use the median and the interquartile range. This already I have taken. So I'll just show one slide to you. Data which we will generate through research are classified into 2 different types. Either we will have a qualitative data and we will have a quantitative data. The example of qualitative data is gender, where you have different category. For example, you have made a questionnaire and whatever the option you have given to choose a respondent, that kind of data will be called as a qualitative data, and quantitative data where numbers are representing. Clear? Either it will be in the form of scale order, or either it will be in the form of ordinal order. I will let you know that what you mean by the scale and what you mean by the order. Slide very carefully. Whatever I have told till now, it is going to explain. On the right hand side, I have written type of data. The type of data is classified into 4 different types basically. The first one is your ordinal, second one is your scale, Third one is your nominal and the ratio. Now, what are the difference between these data, ordinal, scale, nominal, and ratio? Ordinal means data will be given in the form of number. For example, your age, weight of the patient, but does not contain any measurement. That means any measurement units are not there. You can say the example of person, how many person or how many students are there in the class. You will say 30, and who got first rank, second rank, and third rank. So ranking order will be called as ordinal order. Whereas the scale type of data is the numerical data only. The data will be represented in the form of numbers, but it contains units. For example, height, weight, systolic blood pressure, HbA1c, BMI, all these things. So any numbers which contains unit are classified into scale data. Whereas the third one we have, it is called nominal data. The nominal type of data is also classified into two different types, and it is also called as categorical data. I have given you the example of gender. So gender is a nominal data where we have a two different options. So for example, if you have made a questionnaire, and in questionnaire a one question is there, and in one question you have four different options. Clear? So where if any respondent is having options to choose, that kind of data will be called as nominal type of data. And ratio is well known, that ratio of gender, ratio of anything. Clear? So the data which you are going to collect from the hospitals, or you are going to generate in your laboratory, are classified either of these type: ordinal, scale, nominal, or ratio. Once you have collected the data, how to choose the level of measurement? That mean, or you have to use the median. I'll take the example of these two. After collecting the data, you have to see that your data is belonging to which group. Either it is numerical type of data, or either it is the categorical type of data. If data is categorical, you don't have to apply any statistical test. Directly, you can use the frequency, count, percentage, or ratio. These are the level of measurement to explain your data in the research paper or in the thesis. You can write that what was the frequency mean numbers, that how many numbers of males was there, how many numbers of females was there. You can write the count, you can write the percentage, and you can also give the ratio of male and female. Clear? So numerical data, here we have to apply the test for the normality distribution to find out that data is normal or data is not normal. Clear? So once we have collected the data, we will see that data is numerical or data is categorical. If data is categorical, by frequency, count, percentage, and ratio. And if your data is numerical, you can apply the test for the normality distribution, and after applying the test for the normality distribution, find out that which measurement of central tendency. For example, if after applying the test there, that we will discuss in further how to apply, your data is normal, you are eligible to apply mean and standard deviation to your data. If your data is not normal for the mean, in that case you have to apply the median, range, or you have to apply the another measurement of central tendency called IQR, interquartile range. Equation is written with the mean. In the same way, the interquartile range the median. So this rule that to numerical data, before choosing the level of measurement, we have to apply a test for the normality distribution. And the test for the normality distribution, we will find out that our or not normal. If normal, apply mean or standard deviation. If data is not normal, apply median and the interquartile range. So once I'll just take from the beginning, like the data. So how to proceed for the effective research? People are asking the question about the sample size. Clear? How much sample we have to collect for the effective research? Sample size we have to calculate always according to the and the area. For example, of sample size and you have collected the data. Clear? So what to do after collection of the data? Data will be filled in a different software like Info, statistical software, SPSS, or the SAS. So once you have filled the data in SPSS, are you ready for the analysis or not? How to find out? That means directly after filling the data in a statistical software, you cannot do the analysis. Clear? You have to see that your data is normal or not. So to check that you are ready for the analysis or not, for that case also test for normality distribution to check the normality of the data. Clear? Now I will let you know the significance of test for the normality distribution. The first significance is it will let us know that we are ready for the analysis or not. How? For example, your sample size is 200 that you got through the calculation, and after collecting the 200 cases or collecting the data of 200 persons, are you ready for the analysis? If you want to know, you have to apply the test for the normality distribution. Clear? So test for the normality distribution will let us know, or it will provide an idea to us that we are ready for the analysis or not. What will happen? If our test pass, we can proceed with the analysis. Normal and our test fails, we have to increase the sample size of our study. That is the rule. But for the time bound studies, for example, students have to submit the thesis. You don't have to take more number of samples. Still, you can proceed with the analysis, but that time the test will be different. Clear? So, test for the normality distribution ways we are ready for the analysis or not. With measurement of central tendency, we have to use uh, to write our results, and which statistical test further we have to apply in our research. This will be taken by the test for the normality distribution. For example, here I have taken, once you have collected the data filled in a sheet called SPSS, I will let you know how to make a sheet in today's lecture only. Either we are ready for the analysis. If you want to find out this thing, we will check the normality of our data. And I want like to tell you one more thing, that test for the normality distribution can be applied only and only to the numerical type of data. You cannot apply the test to categorical data, for example, gender or the education of the patient. You can apply the test for the normality distribution to age, value, which is written in the form of numbers. Clear? So once you will apply the test, it will suggest you that which statistical test you have to apply further to explain your results. For example, if you are finding a correlation of A with B. Clear? So to justify your research that A is causing B or smoking is causing cancer, which test will be applied? That is based upon the test for the normality distribution. If your test passes, you have to apply the parametric test. You have to apply the non parametric test. Yes, I told you about the significance of test for the normality distribution, and given the example of A is causing B. Now we will discuss the different variables and the coding in SPSS. So whenever we conduct a research, we will have a two different kind of variables, A and B. A will be called as whereas B will be called as the dependent variable. Independent variable, also called as experimental variable, variable, whereas the dependent variable will be called as outcome variable. So if you're conducting a research, your aim of the research objective that A is causing B, we have to see the effect of independent variable into a dependent variable. Clear? So how to choose the test? This we can find out that what is the independent variable in our research and what is the dependent variable. Clear? So I have given the example to you that A is independent variable and the B is dependent variable. Few example I have taken to explain. For example, if you are conducting a study on a hypertension, so dependent variable will be the blood pressure, body weight, pulse rate, or other lab values. For example, if you're taking a blood pressure, so you have to make it two different columns. One is for systolic blood pressure and one is for the diastolic blood pressure, which will be written in the form of number. So both independent variable and the dependent variable can be either numerical or it can be categorical. Clear? Whereas the independent variable, the examples are drugs. For example, you are taking ACE inhibitors, beta blocker, calcium channel blockers, or diuretics. I have taken the example of these four. So out of these four drugs, which one is more effective for decreasing the blood pressure in a hypertensive population? If you want to find out, then different drugs, dependent variable, which you want to evaluate by seeing the effect on the blood pressure. Clear? So dependent variable will be blood pressure, systolic blood pressure and diastolic blood pressure, whereas independent variable which you want to evaluate, that is your drugs A, B, and C blocker or the calcium channel blocker. So these are the examples of independent variable or dependent variable. So whenever you are conducting a research, try to make a list that these are my independent variables and these are my dependent variable, so that you will have it is going to help you the proper statistical test which is best suited for your research. So I will let you know that how to make a sheet in the SPSS. It's a statistical package for the social science. Different options are there. Whenever you will open the software, it will be looks like a Excel sheet only. Clear? So you have to modify the sheet as per the requirement of your research. For example, if you have taken a 24 or 25 variables in your research, I just name out. One is your age, second is your gender, third is your systolic blood pressure, fourth is your BMI, fifth one is your systolic blood pressure, like that. Here, of the variables you have to add, and the characters which you are going to write for the variable names should start from the ABCD. It should not be start from the numbers, and it should not be more than 8 characters. For example, systolic blood pressure, make a short name SBP. If you want to write a body mass index, write BMI. If you want to write age, you can write age because age contains only 3 characters. If you want to write lipid profile, so you can write LDH or HDL. Clear? So name of the variable which we are going to write in the SPSS sheet, that should not be more than 8 character. It should always starts from the alphabet. It should not be start from the numbers. Clear? Now, why we are limited to only 8 characters? For example, whenever we are going to proceed with the calculation, all the calculation we are not going to do with the one software called SPSS. Most of the things we will do with other software also. For example, GraphPad Prism or the EpiInfo or Microsoft Excel. So you have to transfer your data from one software to another software. So for example, I'm taking my data from SPSS to Excel. So if the character is more than 8, the name of the variable will not be shifted to the another software or to the another portal. Only data will be shifted. So keep in your mind that the name of the variable should be less than 8 characters. Clear? And whenever you are going to save your file, for example, once you have completed your work today and you want to save your file, always save your file in a format called .csv extension, that is comma separated value. Clear? Now what was the benefit of this .csv? .csv can be open in any platform. You can open this file in Excel, you can open this file in SPSS, you can open this file in any other software which are used for statistical analysis. Clear? If you have chosen .sav, that is the extension for SPSS, so it will be only open in SPSS, and the size of the file of .csv is very short. Clear? So what I recommend all the viewers, that keep your file name as you like, but file will be saved in the form of .csv extension only. So this is the overall look of the SPSS sheet. I will run the software also, live demonstration, but this is just a screenshot. In this software, you can see on the top you have different options which you can use for the statistical analysis, and different columns and row you can find out. Serial number starting from 1. So here 1 is representing, this one is representing to patient number 1 or the sample number 1, whereas this 2 is representing to sample number 2 or patient number 2. Clear? And on the top you can see different options which we are going to use for our research. Now before I'll proceed, I'll let you know that how to make a sheet for the SPSS. At the bottom of the sheet there is two options called data view and the variable view. Now what it means, data view is used to enter the data once you have made the sheet according or the need of your research. Clear? Data view is used to enter the data, whereas the variable view is used to make the sheet. For example, if you want to design the sheet for your research, you have to switch to a variable view. Currently, whatever the picture you are seeing right now, this one is the data view you are seeing, and this one is the variable view. I have changed the view. Now you can see this one is the variable view. So data view is used to enter the data, whereas the variable view is to make the sheet. For example, you are programming your sheet as per the need of your research. Clear? So different options will be given by the software. For example, in the data view, when you are finding serial number 1, that is the patient number 1, whereas in variable view, serial number 1 will be the first variable of your research, whereas second serial number will be the second variable in your research. For example, I have taken a example of 5 variables. The first one is the age, second gender, blood pressure systolic, pulse rate, and date of admission. Clear? So this name which you are writing in the SPSS, it should be written in the characters. Clear? And the alphabet should not be more than 8 characters. So directly you can write that first variable is your age, second is your gender, third one is your blood pressure, fourth one is your pulse rate, and fifth one is your date of admission. Like so on, you can write all the variables which you have in your research. In most of the research, we will have a 30 to 40 variables if you are working on a patients because of different lab values. So after once you have written the name, second thing the software is going to ask you that what is the type of the variable. Clear? As we know that age is a numeric variable because we have to enter the data in the form of numbers. So we will choose it's a numeric variable. Whereas for gender, which is a categorical variable, we will choose a option called string. String is the option where which we have to choose in the type for any type of the categorical data. Here, for example, literacy rate of the patient, either he is from the rural area or from the urban area where we have two different categories, I will choose string. So for all numeric variable, always choose numeric, and for all categorical type of variable, choose string. The third one is the width. The software is going to ask you that how much space you require to enter the data. For example, age, I have to write 2 characters. For example, my age is 37. I will write 3 and 7. So it contains 2 characters. So we have to choose here 2. For example, if I have to choose the width for the gender, I have to write here 6 because maximum alphabet I am going to write here for the female. Females contain 6 characters, whereas male contains 4 characters. So I will choose here 6. For systolic blood pressure, if I have to write, I will choose 7 to 8. But by default, the software will take this one as a 8. If you want to choose, you can choose. The next one is the decimal. In the decimal, if you want to write, for example, weight, that after the decimal, how many characters you need. Clear? So that you can choose that after decimal you need 4 value or you need only 2 value, that you can choose from here. Now the fifth one is the label. Here things you are going to write, the same it is going to represent in your results. The software is going to make a table for you. The software is going to make a graphs for you. So what will be the header of the table, what will be the footer of the figure, the same which you are going to write here. Written here age in the name, so in the label you have to explain age of what. So you can write respondent. In gender, the same I have written that gender of the respondent, blood pressure of the respondent, I have written systolic blood pressure. You can write pulse rate of the respondent and the what was the full form of things which you have written on the option called name. For example, I have written PR here, PR representing to pulse rate of the respondent. So short form will be written in the name, and what were the explanation of these forms are there can be written in the labels. Now after the labels you have to do a coding, because in SPSS we have to give the codes, and the codes can be given in the option called value. For example, I take the example of gender. In gender, we have two different categories. The first one is second one is the female. So to male, give a code 1, and to female, give a code 2, and that codes can be given in option called value. I will give the live demonstration, and in that live demonstration, I will let you know that how to give the codes. Clear? So after once you have given the codes to your variables, for example, I'll give one more example to you. You are writing about the drugs. So multiple drugs are there in your research, and for example, we'll take the example of drugs used in blood pressure. So to AC inhibitors, you can give code 1. To beta blocker, you can give code 2. calcium channel blockers, you can give code 3, and to diuretics, you can give code 4. So whenever you are going to enter the data in SPSS, you don't have to write the names again and again, calcium channel blocker, AC inhibitors, beta blocker. Directly, you can write the codes that code number 1 drugs has been prescribed to a patient, or code number 2 has been given. You can make the combinations also. For example, the combination of ibupara is there, clear, which contain to 2 drugs. You can give a code number 5. A combination will be given to a patient. So code number 5 can be used. So what I suggest, that first use a codes which contains only single drugs. Once you finish with that, then start with the code which contain 2 drugs. Then once you finish with the 2 drugs combination, then come to 3 drugs combination and the multiple combination drugs. Clear? So value, I will let you know in the live demonstration of SPSS that how to give the values. After giving the values, the option is missing. It is not necessary that we will get all the variables while collecting our data. For example, there will be a missing of data. For example, you have a 200 patient in your research, or 200 animals in your research while doing the pharmacology experiment, and any animal died due to any reason during the experimentation. In that case, the SPSS software will give an option to you that in that case you have to choose a option called missing. For example, I'll take one more example. If you didn't get the age of any patient, clear, so you don't have to write anything, you have to keep that column blank. Clear? So all the columns will be blank, kept blank, where you didn't get the data. Clear? For those particular patients. Finished with the filling of your whole data in SPSS sheet, then a command will be applied in SPSS for the missing value options. Clear? So that all the missing value will be replaced by 999, 999. Now, what is the meaning of this 999? Any researcher who is using SPSS software, they know that digit which will represents the missing value. Now, why we have the missing values in our sheet? Because whenever we are doing a calculation, will not be included, and remaining data will be taken as a 100% by the software. Clear? So this command, okay, how to apply the missing value in the further classes. After that, we have a column, space, that how much space you require, like a width only, to write your data. Much important, you have to keep your data on left side, right side, or in the center in a column. The most important is measure and the role. If you have chosen these two options wrong, then the software will show error to you whenever you will apply the statistical test. That means if your data is ordinal and you have chosen nominal, the software will show error while applying the any statistical test. Clear? Like here, age does not contain any unit. So I have chosen a ordinal. Weight of the patient, then I should choose here scale variable. So 3 options will be given by the third is not visible because I don't have the data. I will show you when I will give the live demonstration of the software. First option I have chosen ordinal, and second option I have my variable name is gender in second serial number. So for second serial number I have chosen the nominal because nominal type of data, and in the gender we have a 2 groups, one is male and second one is. So measure you have to choose very precisely, otherwise the software will show the error to you. All the numerical variable, 2 options are there, and you can choose the scale. Choose ordinal for those which does not contain units. For those numerical variables which contains the units. Nominal variable and categorical variable option will be chosen nominal. And the last one is the role. That what was the role of your variable in your research. For example, I have taken age. So age in my research is acting as what? As I told you, in research we will have a two different kind of variables. The first one is independent variable and second one is the dependent variable. In your research is acting as what? Independent variable or dependent variable? If it is independent variable, input, and I have chosen age into input, also into input, whereas uh, blood pressure like systolic blood pressure and the pulse rate I have chosen both, because I can explain this one with a few example. If I am to compare BMI and the HbA1c, that what will be the effect of BMI on HbA1c? In that case, independent variable and HbA1c can be the dependent variable. Design another model where BMI can be the dependent variable. In that case, you can keep this one as a. So the last option I have chosen none. None means date of admission. I'm not going to use. It is not my independent variable and the dependent variable. It is just a date. So for the date, I will choose a option called none. Independent variable, choose the option called role uh, input, and for dependent variable you can choose the output. But few variables are there which will act as a independent variable as well as dependent variable, model to model. For that case you can choose the option called. So like this you have to make your sheet. You have to add all your variable in the research. That means if you have a 34 variable in your research, you have to add all 34. Then after making the sheet, you can start adding the data. I will show you a few example of the sheet that which I have made. You can see this one is the screen direct live demonstration of the SPSS software. Now you can see this one is the data view at the right hand side, and this one is the variable view. While making the sheet, we have to switch into a variable view. For example, here I have shifted, and here I am going to write that name of first variable age, then name of second variable will be gender, name of third variable will be systolic blood pressure, fourth one is our BMI, and fifth one I'll take pulse rate. Okay, so by default the software is taking this one as a numeric type all, but you have to choose these things according to the variable. In the type option, there is a blue color bar. If you click on that, it will show all the different options which comes under the type. Here, like age is a numeric variable, so I will choose numeric only, and I will click OK. Whereas if I click on gender, in gender, as I told you, the option will be chosen string, so I will choose the option called string on the gender. In systolic blood pressure, I have to enter the data in numbers, so I will choose numbers. In BMI also number, and pulse rate also number. So width I will reduce, I will keep 6. Systolic blood pressure I'll take 4 or 5. BMI sets and pulse rate. I have to write 3 digits, I'll write 4. Clear? We don't require any data in decimals, so I'll make it 0 to all. Now, labels you have to write very precisely. As I told you, if your label is written wrong, so while writing the thesis, the same thing will be shifted to your thesis. So what we have to do? We have to write age of the respondent. In second column, we have to write gender of the respondent. Like so on, you can write for the other variables: age, gender, systolic blood pressure, BMI, and the pulse rate. Now, the value is very important. In value, you can give a code. For example, if I am writing a value number 1, I will write male, add, and in value number 2, I'll write female, and then add. Like this, coding will be done. I'll show screenshots to you if you want. Yeah. So the same thing which I have explained here, the name of each SPSS variable is given. File must be unique, clear, and it must start with a letter, and it may have up to 8 characters, including letters and number. But in the starting, we will not use the numbers. In second, we have a type, as I told you in the sheet, as I shown you in the sheet. The two basic type of variable that we will use, numeric and string. Numeric variable may only have number assigned, whereas a string variable can be used for the categorical variable, where we have to, for example, and string can be used for those variable also, where we have to calculate the mean variance or the standard deviation. Measures, as I told you, are classified into 3 different types. That is your scale, ordinal, nominal, and the categorical type of data. Scale variables are those which contains the measurement. Here, for example, centimeter, weight in kg, height in inches, and so on. Clear? And the ordinal variable, which will be given in the form of number, but does not contain any units. Clear? And nominal will be called as categorical variable. Here is the example. Examples are region, gender, pin code, and the religion. These are the examples of different measures which we will use in the research. This is screenshots. I was choosing the type. So type numerics will be chosen for numerical variable, whereas the string will be chosen for the categorical variable, and the string can be also chosen for other variable where we have to calculate the mean, SD, and the median. Okay, which I told you, labels you have to write very precisely. That is the description of the variable. Value you can give, for example, 1 to male and 2 to female, whereas the missing value, it's optional. You can keep your column blank, but while doing the calculation, you have to take the cumulative percentage instead of the normal percentage if you have kept the column blank. If you have given the commands for the missing value, then you can take directly percentage which will be given by the software. Either missing value will be represented as 999. Here, column is the space which you require to write your data. So here is the screenshot. Okay, how to explain your categorical data with the help of statistical software. Clear? Once you have made the sheet, I will let you know that how to make a sheet in the further classes. There is a option called analyze on the top. You have to get the data for the variable which comes under the category order. So first of all, you have to click on the analyze. A window will appear. In that window, there is an option called descriptive statistics. If you click on descriptive statistics, a new window will open, third one, and there is an option called frequencies. If you click on frequencies, you will find this window we used to call as a frequency window. Clear? And in frequency window, you can see here two big boxes are there. Left hand side, it will show you all the variable which you have made for your research. Clear? And on right hand side, it will be the blank, and you have to shift your variable for whom you have to do the calculation. Clear? For example, gender is the categorical variable, and I have to find out the percentage and the measurement of central tendency of gender, or I have to describe my gender. Clear? How to get the data? So gender will be taken from left side to right side, and click on statistics. And here you can see on the screen, I have already moved the variable from left side to right side, and click on the statistics. If you click on the statistics, choose the option that you require, what you need. You require data in the form of frequency, percentage, or ratio. You can choose, and after that, click OK. If you click OK, you are going to get a table. Clear? Two tables will be given by the software, which will give the data of categorical variable. For example, we used to call as frequency statistics. Clear? The example which you are seeing on the screen is the data of 150 patients, which shows that no data is missing. That missing value is 0. In the next table, you can find a multiple columns. In the first column, you can find that two different category of gender was there. The first one is female, and second one is the male. In second column, it is going to give you the frequency, that out of 150, 83 were female and 67 were male, and the percentage along with that. Yeah. So if you have a missing value, then you have to take the cumulative percentage. If you don't have missing value, then we can take the simple percentage only. Clear? And how to write the results of the descriptive statistics? You can write the data of male and the female are represented in a table shown below, and you can show the percentage. You can write the percentage. The female were found to be 55.3%, and in the bracket you have to write n is equal to 83. Now here small n is representing to the number of females. Clear? Of the study population. That means out of 150, how many females was there? So here I have written just only for the females. No need to write the data of male in the description or in the research paper when you are writing a text, because already you have written, your table is showing the percentage and the frequencies of male in the table. So I have seen different research papers where authors has given the table of descriptive statistics. They have made the graph of the same data, and the same has been written in the text also. So it is not necessary that we provide our data in 3 different forms. only one form is sufficient. Either write your data in the form of text, either give the graph. If not, then you can give the table. Clear? And few research paper I have seen, or multiple research paper I have seen, where I have found that to describe the mean, a separate table is there. To describe the median, there is a separate table, and to describe the percentage, there is a separate table. So it's not like that. We have to use the superscript, for example, hash tag, star mark, or any other superscript to explain that either it is a mean, median, or the mode. I'll show you in the next table. You can see here. Here I have written the data. On the top, I have written mean and SD. But at the bottom of table, I have used the superscript that which data is in the form of number and percentage, and which data is in the form of median and the interquartile range. On the top, it will be mean and SD only, because most of the data in this table is representing to mean and SD, but few things are in percentage and few things are given in the form of median and the interquartile range. Clear? So age I have written 37.6, it's a mean, and 7.6 is a standard deviation. Clear? Whereas male 67.0 and 44.7, and I have written small a. You can see here a small a is written in a superscript. That means a is representing to a percentage, whereas small b is representing to a median and interquartile range. So this 5.6 and 1.5 is not the mean and standard deviation, it's a median and the interquartile range. Now, why I have written glucose in median, the value of glucose in median, and the value of glucose in interquartile range? Why not mean? Because when I have applied the test for the normality distribution on the data of glucose, and my test has given the idea that data is not normal, so I have chosen the median as a measurement of central tendency to explain the glucose, not the mean. Clear? So by applying the test for normality distribution, you can find out that which data which will be given in the form of mean and which data will be given in the form of median. So a single table can be made for the descriptive statistics. There is no need to give separate table for male, separate table for female, separate table for income, smoking, and BMI. All can be clubbed in a single table. On the top, you can write the measurement of central tendency, which is most common in whole variable, and remaining can be added in a superscript form. Clear? So like this, you can make a combined table of descriptive statistics for your research paper. Now, I will let you know that how to find out, and I'll take the first test, which is the mother of all tests, the test for the normality distribution, for a study which contain numerical variables, and how to find out either our data is normal and or not normal. These are the commands. You can take a screenshot of this one to SPSS. So what are the commands? First of all, you have to click on the analyze. A window will open. Then you can click on the descriptive statistics. A new window will open, and explore is the command. Explore is the command for test for the normality distribution. You will not find any word called normality distribution in the SPSS software. So by using explore command, we can run the test called test for normality distribution. Clear? So here I have taken the example of age. For example, I have to find out either the data of my age is normally distributed or not, because uh, I have to choose the measurement of central tendency. That age I have to write in the form of median or the mean. Clear? So software will give multiple things to you, or 5 different ways are there to find out that our data is normal or not normal. So these commands will be followed. We have to plot a graph also. We have to plot a histogram. Then we can click continue and OK. I will show the screenshots to you how to apply. This is the slide which shows how to find out the normality of a numerical data. Once you have filled the data in SPSS, now how to fill the data in SPSS? Like you will fill the data in Excel sheet, you can fill the same way. You don't have to give coding for the numerical. You can see here, I have entered the data of age of the patient at 46, 36, 36, 46, 36, 33, 55, 34, 31. So each row is representing to each patient. So you can see here the data of I have added in this sheet around 150 patient, but screen is showing only 18 patient that in yellow color. So like this, you have to fill the data of age of all respondent of all the patients in your sheet. Clear? So once you have added the data of age, you are ready to find out that your data is normal or not normal. Because if you want to correlate the age with any other variable, for example, you want to find out that which drugs are prescribed more under which age group. You can find out with SPSS, and multiple things are there. So many options. So here we are talking about the measurement of central tendency. That if I got the data of age, I should write mean or I should write the median. So how to do? As it shown the commands in the last slide, I'll repeat again. You have to click on analyze. Then you have to click on the descriptive statistics. If you click on descriptive statistics, it will open a new window. This is the command for test for the normality distribution. Clear? If you click on explore, this window will appear. Now in this window, this is the window of explore. You will find left hand side all the variable which we have made for our research. Clear? Age is mentioning in the dependent list, because already I have shifted from left hand side to right hand side. Clear? So here age is acting as a dependent variable. So by using this icon, a small tab box, you can switch the variables from left side to right side. So age should be moved into a dependent list box if you want to find out the normality of age. Clear? So once age will be shifted to a dependent list box, you can click on statistics. Here, see, you can see already I have moved the age into a dependent list box, and you can click on the statistics to choose different options. And if you click on statistics, this window will appear, uh, statistics of explore window. And by default, the confidence interval, it is taking 95%, and the margin of error is 5%. The software is taking by default 95. So if you have chosen while calculating the sample size of your study, the confidence interval 96 or 97, you can change here. The higher the confidence interval of the study will have the more impact, and the chances of error in the study will be less. For example, for my PhD, I have chosen the confidence interval of 97%. So chances of error will be 3%. So standard p-value for my study will be 0.03. And one more thing with this test is there, whenever we will get a table of results, the p-value for this particular test will be taken opposite to all other tests. For example, for all other statistical tests, we will find significant if our p-value is less than 0.05. But for this particular test, we will find test is passed if our p-value is more than 0.05. So 5 different ways are there, by which mean you can find out your data is normal or not normal. How to do it? Once you will check that confidence interval of your study is 95%, click on continue. If you click on continue, it will show this window to you. Sorry. You have to click on plot. In plot, you have to choose a few options. The first option is your histogram, and second option is your normality plot with text, because you have to plot a histogram also, and you require a normality plot to find out that your data is normal or not normal. And it will give a tables and graphs, by which mean you can find out your data is normal or not normal. If you click continue, SPSS will give the output sheet. Output sheet here represents the data which you will generate up after applying the statistical test. For example, if I click continue here, I will get a sheet and the results of SPSS test for the normality distribution. So how the results look like? The first one is the case processing summary. The case processing summary will give you a descriptive statistics of age and the capital N. How many numbers of patients or animals was there in your research? It is showing that N is equal to 150, which I have used to generate this data that contains 150 patients, and none of my patient age was missing, so it is showing missing 0, and total is uh, n is equal to 150 and the 100%, that we have filled the all the patients age in the SPSS sheet. Clear? Now here is the example of descriptive statistics made by the software. You can see the mean of the age. The software is giving 37.62 years. Median, the software is giving 37.50. Somehow similar, little bit difference. It is giving variance, standard deviation 7.6. Minimum age was found to be 18. Maximum age was found to be 56. Range is 38. And the interquartile range is 10. And here you can see the value of skewness and the kurtosis. Now, what do you mean by the skewness and the kurtosis? Skewness represents the data which is on the left hand side or on the right hand side in the graph, and kurtosis will represent to the height of the graph. So as I told you that 5 different way are there to find out that your data is normal or not normal, and how to find out that I'll show you in the results. But how the results will be written, I have already applied the test and I know that this data is normally distributed, so I will use the mean for this one. There is a two different formats are there to write the results, well known, which is accepted by most of the journals. The first one is the MLA, that is Modern Language Association, and second one is your APA, American Psychological Association, which will give you the basic text, by which mean you can write the your results directly. You cannot write that male equal to 37 and female equal to so much. Clear? So what we used to do, we have to use the standard formats to write our results. Either you can use the APA format or either you can use the MLA handbook 6th edition formats to write your results. So how the results will be written? You can write the results here shows that the mean, in the bracket you have to write SD is, and then you can write the mean 37.6, and standard deviation can be written in the bracket 7.6. Unit you have to write years, and if it is your if you are writing about median, so instead of mean you can write median is 37.5, and in the bracket you have to write the interquartile range, which is the complement of standard deviation. So always write standard deviation with the mean and interquartile range with the median. And you can also provide range that age ranging from 18, which is the minimum, and 56, which is the maximum. And you can write the value of skewness and the kurtosis. The skewness was found to be minus 0.258 and kurtosis is found to be minus 0.198. Now what the value of these skewness and kurtosis represents, I will let you know. First, I'll let you know that how I have found out that my data was found normal. Two well-known tests are there, and SPSS is also using these two tests. The first name of the test is Kolmogorov, and name of the second test is Shapiro-Wilk. These are the two scientists who has developed the test for the normality distribution. In the test, what we have to see? We have to see that our significant value or the p-value should be more than 0.05 for this test. For all other tests, we are taking p-value less than 0.05, but especially for this test for the normality distribution, it should be more than 0.05, which is the standard. So here I am getting the Kolmogorov value 0.200, and for Shapiro-Wilk I am getting 0.195. Here, if any of these two tests, Kolmogorov or Shapiro-Wilk value is more than 0.05, you can say your data is normally distributed and you are eligible to apply the mean for your research. Here, so this is the first rule. By seeing the value of Kolmogorov and the Shapiro-Wilk. Clear? So this table represents the results from two well-known tests of normality. Now, which test we have to apply? Shapiro-Wilk test is most appropriate and can be used for the sample size where the sample size is less than 50. Clear? And Kolmogorov can handle the sample size up to 2000. I'll repeat again. Shapiro-Wilk can be used if your sample is less than 50. For example, person are doing animal study where the sample size is less, 30, 40, they can use the value of Shapiro-Wilk. And if the person from medical background, doctors, pharmacists, pharmacy students, they are using large number of samples of the patients, so they can use the value of Kolmogorov. But if your sample size is more than 2000, then we have to see our results with the help of graphs. Then the value of Kolmogorov and the Shapiro-Wilk cannot be used. Clear? So for sample size less than 50, use the value of Shapiro-Wilk, and the sample size up to 2000, use the value of Kolmogorov. If sample size is more than 2000, we have to see our results with the help of graph and with the help of the value of skewness and the value of kurtosis. Next slide. You can see here, this is the formula for the test for the normality distribution. We used to call as Z score, x minus mu divided by standard deviation. Clear? So what we have to see, our Kolmogorov value and the Shapiro-Wilk value should be greater than 0.05. That means our data is normal. If our sample size is more than 2000, then software will plot a graph to you, three different graphs. One is your normality plot, second is your histogram, and third one is the QQ plot. So by seeing the QQ plot also, you can find out your data is normal or not. You can see here, if data points are closer to a diagonal line, it means your data is normal. And if you see your data is on in this area or far from the diagonal line, you can say your data is not normal. And which method you are using to evaluate your data for the test for the normality distribution, the same thing you have to explain in your research paper, that by taking the value of Kolmogorov, by taking the value of Shapiro-Wilk, or by taking the value of QQ plot, histogram, or normality plot, by giving a reference that by which method you have evaluated that your data is normal. So if data points are closer to a diagonal line, you can say your data is normal. This is the picture of histogram. If now, if you find your histogram is taking a bell-shaped curve, you can say your data is normally distributed. If your data is not normally distributed, it means it have a skewness in the data, either left side skewness or the right side skewness. Here you can see in this picture that histogram is taking a bell-shaped curve. Clear? So until and unless I will not fit the normality line in this graph, I used to say this one as a histogram only, because all bars are attached to each other. Clear? So you can see that minimum age we have found 18 and maximum age was 58. So data is ranging from 18 to 58, and maximum uh, patients or animals were age between is a patients only, and age of the patient years is mean which we found is around 36 or 37. So you can see the maximum peak on the center. So if we got the bell-shaped curve, we can say a data is normally distributed. So until unless I will not fit the normality line in this histogram, I will say this one as a histogram only. So if I'll fit the normality line, we can say this one is a normality plot. So how to do it? You have to double click to fix the normality line on the graph. You have to double click on the picture, and on the picture you will have a icon for the double click to activate the graph. And if you activate the graph, here is the icon which show the distribution curve. If you click on the distribution curve, you will have a curve like this, and you can find out that your curve is taking a bell-shaped curve or not. In tomorrow's class, I'll take this test live. I will not explain through PPT. So we have to see that either the graph is taking bell-shaped curve or not. If it is taking bell-shaped curve, that means the data is normally distributed. Now here you can see the example of two different graphs. On left hand side, I'm getting bell-shaped curve, which is the example of my study. And on the right hand side, you can see the data is having a skewness. That means data, most of the uh, values are on the right hand side. So what I will say, that this graph is a positive skewed distribution of glucose. So this graph will be taken as a normal distributed data, and this graph will be taken as a not normally distributed data. So my either the height of the graph, height of the bars will be on the right side. You can say this one is a positive skewed graph, and if the data is on the left side, maximum bar having height on the left side, you can say the data is negative skewed. So if you got the skewness in your data, you can say that data is not normally distributed. And for this graph, you can say that data is normally distributed. So for normally distributed data, you can use mean and standard deviation for the graph which you are getting a bell-shaped curve. If you're not getting a bell-shaped curve for a data which contains skewness, either positive, negative, we can say left side skewness or the right side skewness. For this kind of data, we have to choose the median as a measurement of central tendency. Here is the example, measurement of asymmetry skewness. Now, what do you mean by normal distribution? Normal distribution means that all the three measurement of central tendency, that your mean, mode, and median, are meeting at a single point at the center. You can see this data we used to call that data is normally distributed, and we are getting a bell-shaped curve. Your x bar mean is equivalent to mode and equivalent to median. Clear? So x bar is equal to median, and median is equivalent to z, that is your mode. So for this kind of data we can use median, and for this kind of data, if you are seeing a half curve like this, this is right skewed, we can use median, and for this kind of data also we can use the median and the interquartile range. So two different graphs which is showing the different curves of the skewness. Numerically, if we want to find the z score, and numerically also you can carry out this test. The formula is z is equal to x minus mu divided by standard deviation, where x is the data value which we are taking. So example I have taken, you can see that a set of economics final exam grade are normally distributed with the mean of 65 and a standard deviation, and by using the formula you can find out the z score, and to find out the p value we have to use a table. This is the table which we are using for to find out the z score and to find out the p value. For example, if we got the z score by using x-axis and y-axis, we can find out that what will be the p-value for the test. And if p-value is more than 0.05, you have to see this row 0.05, because the confidence interval while applying the test we have taken a 95%. So cutoff point for the study will be 0.05. So on the 0.05 you have to compare your value with the standard value with the z score. For example, if you're getting a z score minus 2.8, and here p value is 0.05, you can compare your value with this one, and you can find out your data is normally distributed or not. The same thing I am going to send you by mails, and we will also upload these presentations in SlideShare and our YouTube channel. In our college website also, there from there viewers can download the daily presentation. Might be it will take time, delay of 1 day or 2 day, but all the materials for this lecture series will be given to all participants. Clear? So by applying test for normality distribution, we can find out that our data is normally distributed or not, and we can choose the measurement of central tendency, that which measurement of central tendency is best suited to my data. So from today onwards, do not use mean or standard deviation like this only. By seeing the research papers that author has written his data or her data in the form of mean or SD, so I will also write my data in the form of mean or SD or the median or interquartile range. Clear? Apply test for the normality distribution to your data. Choose that which measurement tendency is best suited for your research. Now I'll tell one more significance is of test for the normality distribution to you. This test will help us to find out that which further test we are going to apply for our research further. For example, as I was talking to you that A is causing B, clear? So if I want to justify that A is causing B, which test will be applied? In statistics, we have two different type of tests. The first one is your parametric test, and second one is the non-parametric test. Parametric test will be applied when data is normally distributed and your test for the normality distribution passes. Whereas non-parametric test will be applied to those kind of data where test of the normality distribution fails, or we can say data is not normal. Clear? So example of parametric test are student t-test, independent, ANOVA, Mankova, and example of non-parametric test is chi-square, Kruskal, Wallis, and the Mann-Whitney U test. Clear? So, for example, if you're finding your data is not normal, and the student have to bind their thesis because they don't have the time to increase the sample size. In that case, still you can uh, continue with your results part, but for the time bound studies you can apply non-parametric test if you don't have time to increase the sample size for your research. Otherwise, what is the rule of stat says? Rule of stat says if your data is not normal, you have to increase the sample size of your population until unless you're not getting a bell-shaped curve. If you're getting a bell-shaped curve, then only you are ready for the analysis or the statistical analysis. Otherwise, uh, still if you want to proceed, you can proceed, but with the non-parametric test, you are not eligible to apply the parametric test for your statistical analysis. Okay, so test for the normative distribution will let us know that which measurement of central tendency we have to use to describe our data. Test for normative distribution also will help us to find out that which further statistical test I have to apply for my research. If test passes, we will go for parametric test. If test says data is not normal, we have to proceed with the non-parametric test. So in this lecture series, you send a mail to me, you send a WhatsApp number, you send SMS, by any other mean of communication, that which kind of topics you want to explore in the further classes, and I should give emphasis on which topics so that I will elaborate those topics uh, in time. Or remaining the schedule for the classes will be mailed to you by evening, that which classes will take which test. Clear? So for the analytical statistics, this I'll take tomorrow also, and just I'll give a background that analytical statistics, the aim is to find a correlation between two different variables. For example, I have given you A is causing B. So how A is causing B? If you want to prove this one, you can justify this one with the help of analytical statistics. That means we have to apply a statistical test, but there is a rule of inferential statistics which I'll discuss tomorrow, and hypothesis also. Before applying the test, we have to make the hypothesis for the test. Each test is having a hypothesis. So by making the hypothesis for each test, I will take the live test and the live demo of all the tests with the help of SPSS software with numerical. Like I'll also show you with the help of these type of calculations which I have made, like how the test will be applied with hand if you want to do it manually, and with the help of the software. So by two different means, we are going to solve all the tests. Either it is student test, independent, ANOVA, one way, uh, two way. I'll teach you by SPSS only because it's a huge calculation. Remaining Mann-Whitney U, Kruskal, Wallis, linear regression, linear correlation, general linear model, factorial analysis, time frame analysis, all these things will take in the further classes. Clear? So see you tomorrow, same time. I will be online at 11 o'clock and waiting for your comments. Thank you. Thanks a lot. हेलो, क्लोज कर दो। कर दो, बंद कर दो।